Hello, everyone. Welcome to today's colloquium. It's a pleasure to have Professor V. Nagaraja visiting us. So, Professor Nagaraja did his bachelor's and master's from Bangalore University and then did a PhD from IISc. He was a postdoc at University of Basel in Switzerland and then at the University of Rochester in the US. So his research focuses on DNA topoisomerases, looks at lots of topology modulation in DNA and also how gene expression is regulated in various aspects of cellular function. He's also interested in the biology of pathogens that underlie tuberculosis, which is what he'll tell us today. So Professor Nagaraja is a legendary teacher. I heard of him when I was a student in IISc long back. He's very well recognized. His work is very well recognized with lots of awards, some of them being the Bhatnagar Award, the GNR Gold Medal, JC Bose Medal, the World Academy of Sciences Medal, and most recently the Sir M. Visvesvaraya Senior Scientist Award from the state government. He's a member of all the three science academies of India and also the World Academy of Sciences. So it's a pleasure to have Professor Nagaraja, and he'll tell us about how to think about resurgent drug-resistant tuberculosis.
Thank you, Krishnamurthy, for this very generous introduction. Actually, I speak loud. So with the mic, if I'm speaking too loud, you may have to tell me I can remove the mic. At the outset, thanks very much to Professor Gopakumar for this invitation. I happened to host him a long time ago in 2016 or 2017 in JNC. When I was in JNC, he gave a fantastic talk, though I don't remember the details I told him. So now he's returning this favor by inviting me over here. This is my first time in ICTS. I'm delighted to see my friend Spenta here and distinguished Professor Ashok Sen here. So I hope I will be able to do justice for the next 45-50 minutes, but some of the biology talk. Let me assure you, I will not go into technical things, but towards the end, I have to also show off that I'm doing some science and a little bit of experiments will come in, but I will minimize the details of experimental detail, any aspects of it. So I chose the topic today, more general one, and which is of course my long-term work, starting from PhD days. I started working on TB problem as a PhD student in Indian science, then went on to something else, different places. There's some kind of echo. I don't know. And then when I came back, I restarted working on tuberculosis. We also work on other things, but what we do in tuberculosis, I'll try to give you a little bit of bird's eye view kind of thing, a few snapshots, but nothing into depth. So this is the second time I'm speaking to non-biology audience. Long ago in Calcutta, I gave a talk in the biology-inspired physics. Somebody invited me there, but there were a few more biologists there, so I was in a little bit better comfort zone there. Anyway, today what we discussed today is about the bacteria. Mycobacterium tuberculosis is the full name. This is an ancient bacteria. The organism is there before humans evolved on Earth. In fact, perhaps it has come from, it can infect many organisms. Almost even invertebrates it can infect. But we have a fish TB, it's called Mycobacterium marinum, and reptiles have TB and others have TB. And the cow TB called Mycobacterium bovis. So there is a evolution of TB bacteria, and in humans, this TB bacteria is found in the Egyptian mummies, the lesions. So it's a very ancient disease. It has co-evolved to infect humans, a lot other than humans, other organisms, other animals as well, and learn to survive the host defense. In the evolutionary arms race with its host, it has found a way to outsmart the host, that is humans. And that is basically the theme of the talk. But I will try to say how we try to manage that disease, how to find a solution. The efforts over 50, 60, 70 years from scientists all over the world to deal with the organism. And on the left is the students who contributed to the work. There are others also, and my main collaborators and the funding. The last funding is called More Medicines for TB, a European consortium. Good times, I had 5 years funding from them in a large European consortium to find new drugs for TB. In fact, we found new molecules. We are still working on them. Right. As I said, it's a very ancient disease, but the problem is now much more serious. Now you have drug-resistant TB, and the drug-resistant TB comes in different flavors. It can be multiply drug-resistant or extensively drug-resistant. And if several years ago in Delhi, in Mumbai, the hospital, they came up with a totally drug-resistant, MDR, XDR, and TDR. The definition of what is MDR, XDR, and TDR, I will not go into today, because it depends on what drugs. Of course, TDR means no drugs can treat them. And those definitions are not necessarily important here. Suffice to say, as such, right now in India, there are more than 20% cases of TB cases are drug resistant. It could be even more. And because our epidemiology and studies are not that necessarily up to the mark. And in fact, definitions are there. But then, when it comes to TB, in almost every family, there is someone who was infected with TB. In fact, way back, my mother, when she was only 13, I think she was infected with TB. She came down with the TB, but she was treated. Apparently, the village doctor happened to be my father, and he married her, of course. Okay, so, but the TB is known to be a lung disease, right? All of us know that people cough and other things, but TB bacteria is so clever, it can infect every organelle of the body, every organ of the body. You can have a brain TB, you can have a skeletal TB, gastrointestinal TB, eye TB, lymph node TB, and all kinds of skin TB and other things, and name a system. And in fact, a few years ago, one of my IISc colleagues had a serious ear infection, and for 6 months, nothing worked. Finally, when the anti-TB drugs were given, it was cured. And in fact, bone marrow transplantation, when the people are recovering, they come down with very high fever. And when they come down with the high fever, nothing works, then they use the anti-TB drugs. That if somebody asks the question why it happens, I can tell you. One young doctor from St. John's wanted to come and work with me about this problem, but when he started planning out the experiments, he went off to US. So we didn't continue that work. Right. MDR and XDR TB, in fact, I must say, almost all of us are infected with TB in India. But we don't come down with the disease because our immune system is strong. People immunocompromised, in variety of reasons immunocompromised, they can come down with the TB. Or there is a heavy dose. Anybody traveling in public transport would come down with this, tuberculosis. I mean, get infected with tuberculosis, but you won't come down with the disease. So most of us, if not all, have a dormant TB sitting inside us. It's a dormant, don't worry, Spenda. It's this. Otherwise, how do you expect the young Swiss, Europeans, Americans come to India, go to trekking in Ladakh or elsewhere? When they go past 60, 70, they had no other occasion or opportunity to get infected with TB, they come down with the TB. That's called TB reactivation. So when you get older, our immunity system is down, they can come down with the TB. And these days, of course, even younger people get affected. Yeah.
This dormant TB that is there That means that it is localized or it is spread out, or
It's a fantastic question. There are hiding niches. Until 10 years ago, we didn't know what are these niches. And as some of I have a few more slides, then it will be clear to you. Even as of today, as I'm speaking, nobody has seen, there are few niches where they have seen the reactivation spots. Okay. And one science translational medicine paper and few others, they have shown in some skeletal muscle, somewhere else, it's hiding points. They are not, when you infect in the lung macrophages, our defense cells, they are not, it's not dormant there. From there, it can get transmitted to elsewhere. And it is hiding there. The dormant TB is equivalent to something like, I was talking to Krishnamurti, there is a bacterial virus called lambda. It can lie dormant. So when it is dormant, it's like inside the bacterial cell, it will not propagate. In the same way, TB bacteria can hide itself. Now we have found 2 or 3 niches where it's dormant. But there are experimental models for dormant TB, but they are close to the reality, but no one has actually visualized the population of dormant TB. But reactivation people have studied. In fact, one of our recent work is we have stumbled on some interesting reactivation scenario. I will not speak about it. So now, TB bacteria treatment is a challenge. From WHO treatment regimen is a 6 months treatment for a drug sensitive TB. That's a multi-drug therapy and it is a long treatment. None of us would like to take medicine even for 7 days when the antibiotics were prescribed by the doctor. After 3rd day, I think we have the habit of stopping it, completing it. That can lead to drug resistance. But imagine in this case, so the treatment regimen is, there are 4 drugs, rifampicin, INX, pyrazinamide and ethambutol. That is a short form. Don't worry about this. Those 4 drugs have to be administered for first 2 months. Then only 2 of them are continued for the next 4 months. That's called direct observed therapy, DOTS program of WHO. This is for drug sensitive TB. And if there is a original TB, like about 80% of the population now, and if the patient follows the patient compliance is there, then I think it's sterilized. It happens. And many people have come out like that. But many times, depending on the background, though there's a free drugs and it is given, in fact, this DOTS regimen is very, very well organized and in South Indian states and only few states in the northern hemisphere, northern part of India. But during COVID time, whole thing got messed up because simply all these healthcare providers of TB were channelized for COVID treatment. And that's why you read in newspapers that there's a TB cases of surged now post-COVID. Right. So this is for, many states they have very good system to take care of this, and the drugs are provided free from the state. Yet our numbers are huge. In the newspapers report one outbreak of one virus or the other one. Of course, pandemic was the big thing. But TB, every day people die of tuberculosis more than any one of these things. Even today, the government plans is saying about eradication initially 2025, now 2035. But I was saying one young colleague a few days ago, when you retire also you'll be dealing with TB. It's not such a simple case, having worked a lifetime on this. So can it be improved? Yes, that's the idea. Can it be improved? That's why we all work in that area. In the drug-resistant TB, though, the duration treatment could be 18 to 24 months. And totally different kinds of drugs. And I won't elaborate on them. And after 24 months, only chances are less than 50% to survival. And it's quite serious. So the other problem is diagnostics. As I said, there's a ear infection or bone marrow transplantation and people come down with high fever. You can't detect this TB bacilli Unlike, if it's a urinary tract infection or some other infection, you can do the culture and you can find out what bacteria is, which kind of bacteria causing, and you can treat with, even if it's drug resistant, you'll find some other drug to treat them. So that's not easy. And then there's a childhood TB now in India. It's more than 15% documented. Very difficult to diagnose, extremely difficult to diagnose. So we are talking about not a small number, huge number, as I said, millions. So it's a tough bacteria. Among the bacteria is tough. It's a multiple fortress, and multiple in the cell wall, cell membrane, everything is much more complex than laboratory bacteria like E. coli or some of the other bacteria, much more complex. And then bacteria is very clever, protected by not only waxy, lipid, and complex array, and it has found a way of countering the all kinds of host attacks, host counterattacks, immunological and chemical attacks. And next few slides, I will summarize this. So just for the to bring you into the theme of the talk, MTB infection normally, let us say, a lot of typical symptoms are a particular kind of continuous coughing, right? And when the patient when the infected individual coughs, lot of bacteria released, disseminated, inhaled in the lungs. The macrophages are our defense cells. They capture all the invaders, whether it's virus, bacteria, anything and everything in the lungs. And now their job is to destroy the invaders. How they destroy? There is elaborate mechanisms. But so they enter the alveolar, means lung macrophages, and inside the macrophages, most of the other mundane bacteria get disintegrated, destroyed by variety of mechanisms. I will not go into the details. But TB bacteria is found to outmaneuver the macrophages and survive. And it does what it does is subverts the macrophage. I have a next slide. I'll show you how it subverts it. Just to give an idea, not go into any details. That itself can be an entire seminar topic, the different mechanisms. And it recruits other kinds of neighboring cells. Infected cells, it recruits host cells and utilizes the host machinery, host lipids and other things for its survival. And in fact, it has a two different kind of lifestyle. It extracellular, when you grow in the laboratory, versus intracellular, it uses different biochemical chart, biochemical mechanisms. So it inhibits inside the host the nitrosoactive or oxidative stress. I will not elaborate what these stresses are, and suppresses the host-mediated, apoptosis is the way cells get killed. So infected cells can be killed by apoptosis. That way, multiplication cannot take place. But it will suppress the apoptosis so that it wants to multiply there. Okay, and in fact, I'm not a cell biologist or a immunologist, but of late I got into this with the recent problem, so started learning a little bit about this kind of phenomena in biology. So basically, the immune response, see, in our defense system, we have two mechanisms. One is called innate immune response. This is all inbuilt, like, whatever in the macrophages and what's happening. Then there is an adaptive response. That is the antibody generated, it comes later. Okay. So this innate immune system, it overcomes. Then when it starts surviving in the macrophages, by manipulating the macrophages, it can get into, where did I leave the pointer? Yeah, here. It's a basically when it overcomes, adaptive immune cells are activated at that stage. But now, if it overcomes adaptive immune response, it progressively multiplies inside this mass of tissue. This is called granuloma. I will come back to this. And then, when it becomes too large, it bursts open the cell, which is already under its control, and that's how the cough and everything is. Whole cycle comes. On the other hand, if the adaptive immune system is quite strong, then TB starts hiding as a dormant bacteria. So TB just doesn't die. Your adaptive immune system cannot kill it. Of course, in many cases, some of the bacteria will get killed. It is tug of war. It's arms race. In arms, there will be quite a few bacteria dying. Now here, what you see is one of the enlarged version of those infected cells. These are the mycobacteria inside the its original macrophage is already lysed, and neighboring cells are taken up. There's about 8 or 10 different kinds of cells are taken up there, and nutrients are coming there for this multiplication of. So this is an anaerobic environment. There it's the bacteria is thriving, and this is here. It's basically is able to thrive, and from there it can go to either dormant state or multiplication state, as I mentioned already. Right. So inside the macrophages, usually the other pathogens, when they come in, they are engulfed into a vacuole. This is inside this vacuole, this is the pathogen here. It gets digested. Different kind of enzymes, lipases, hydrolases, proteases, they can digest. Then that can lead to start, their antigen response. So next time when the virus infects, you have the antibody. That's because of the adaptive immunity is taken over. Here, what Mycobacteria does is, this vacuole and this fusion it prevents. As a result, it doesn't come in to get digested. It stays there and starts multiplying by variety of mechanisms. There are already 30, 40 different mechanisms how it's able to out maneuver the macrophages. I will not go into the details. So just to recapitulate in just words, 1/3 of the world population carries latent TB infection. This is okay. No available drugs can kill latent TB, the dormant TB. All these drugs treat active TB. And Mycobacterium tuberculosis becomes dormant and phenotypically drug resistant. Under those conditions, you can't treat with. So it's drug resistant because no drugs can act at this stage, and it can survive from any kind of host stress. And then most dormancy models use single stress factors, and they can't really recapitulate. But all of us use these dormant models when you have to study the bacteria. I mean, whoever is working on this kind of thing. And you need much more things to understand. There are multiple models about this. So it's an ancient bacterium. Yeah.
It has so many ways to multiply, right? Like, so why does it choose to stay dormant in a big majority of the people?
The majority, because as I said some time ago, see, if it is, if it is able to, it's let us say 100 bacteria infect the host. Some of them will die. But some, depending on the state of the individual, what can happen is, inside the macrophages, you will have TB bacteria multiplying. Alternately, as I showed here, this is the initial innate immune response. If it overcomes, then it is an adaptive immune cells situation. Under these conditions, these adaptive immune cells can be activated. When they are activated, you have two options. If they get the upper hand, then it goes and hides. It goes and hides here. Controlled establishment. It's a latent infection. On the other hand, if it's a adaptive immune response also is not able to cope up with the bacteria, then it proliferates. So it has basically two kinds of alternate modes. Okay, and by itself, it has a lot of details built into this. There's specialized meetings only on adaptive immunity after TB infection, or how initial interactions, etc. Right. So did I make myself clear? So has found ways to survive, can stay dormant inside the healthy individual, difficult to handle. Yeah, and there's another important point is the HIV and TB are great friends, great nexus. A lot of HIV patients die of tuberculosis, more than 50% of them. Because they are immunologically compromised, then this latent TB can be activated. And dealing with HIV TB co-infection is a very huge challenge. Though HIV by itself is more controlled than TB, this is a challenge. So multiple strategies are required. So there are 6 components one can think of for dealing with the TB. One is pursuit of high quality DOTS expansion and enhancement. Now, this is a DOTS, that's a direct observed therapy, 4-drug regimen I talked about. Address TB, HIV, MDR TB and the needs of the poor and vulnerable populations. Control the health system and primary health care providers, taking care of the engaged healthcare providers and whatever the way it is done. Empower people with TB and communities through partnerships, and people like me come in to carrying out research, and if the government enables that kind of research. Right. So how do you deal with the TB then? There are obviously 2 approaches. Either you can have vaccine or drugs. Both approaches are tried, but when you talk about a vaccine, so TB being a much more complicated infection, and even today the drugs are effective. It's a prolonged treatment, but still you can cure TB, as I said. But no vaccine has been magic vaccine. Almost all of us are given BCG vaccine. BCG is a attenuated TB bacteria. When you are very young, we are given this. Its efficacy is very poor in Indian population. But we have the innate immunity because we are naturally infected with TB. It's not just because of vaccine. On the other hand, it is more effective in Caucasians, but yet it is not most effective. So but there is even recently, last week I was in a TB conference. It says it's making a kind of a foolproof vaccine, unlike for viruses, is a challenge for TB because of the kind of infection, systemic infection, way it can infect so many different ways, etc. So there are 6 ways of when people have thought of making vaccines. You can have vaccine to prevent infection, a conventional protective vaccine, that's what we normally give for various things. And then you can have a therapeutic vaccine, or you can have a booster vaccine, or facilitate chemotherapy by vaccination, and vaccine to prevent reactivation. So these strategies are tried, and I will not go into this again, and multiple approaches. And this table, I'm not going to go through. There are multiple, each strategy, so many vaccines at different stages, and some show more efficacy. Yet, a vaccine is not a good strategy, in my opinion. Of course, there are people who still pursuing that vaccine strategy. Simply because the vaccine strategy, even if you get a new vaccine, its efficacy you don't know. By the time it gets in, it will be 20 years. And its efficacy, from whatever we know so far, it is not going to be as effective for a variety of reasons, as I said. So why do we need TB drugs then? Because existing drugs have drug resistance. So we need first-line TB drugs are more than 40 years old and must be taken for 4 to 6 months, and treating drug resistance TB usually takes 18 months. Excuse me. Fast-acting drugs are needed. So anybody who can find a magic drug used even for 2 months, they're potential Nobel Prize winners, simply because there is such a major problem. That has not happened. But having said that, there is a new regimen at this moment, the 3-month regimen for the persistent efforts of a 3-drug combination, which is already in the WHO as prescribed in case of TB. So emergence of drug-resistant tuberculosis, so you need new drugs. And of that, as I mentioned, MDR, TDR, etc. And treatment against latent TB infection, that's again a challenge, as I mentioned. If you can eradicate, for example, some of the viruses like smallpox and all, we claim to have eradicated simply by effective vaccine treatment. And polio, by and large, if it is eradicated. So viruses can be contained. And even COVID has come and gone. Now it is endemic and it is not going to be such a serious threat. But there can be something else, some other zoonotic or some other infection can take. So this again says slow progress in vaccine development, and even after development there is no guarantee because TB is a systemized. So there are new drugs, there are landmark papers appearing, and this is already in the phase 3 clinical trial. This is already a wonder drug. Serendipitous discovery by Janssen Pharmaceutica. They are looking for something else, and the energy ATP synthase, ATP inhibitor, diaryl quinolines, they have come as a bedaquiline. That's one of the combination drug, magic bullet at this moment being used. This is the effect of multiple multi-group partnerships. So I was part of one of these groups called More Medicines for TB. Same group, European Union, had new medicines for TB. Afterwards, more medicines for TB. And now they have ERA TB, eradicate TB. So this multicentric, some 20, 30 groups come together to do work. So we have developed many strategies. Over the years, I have been working on TB bacteria. Basically, what we do in the laboratory is trying to understand the bacteria. This is a complex bacteria. We do biochemistry. We do microbiology. We do so-called molecular biology. What we try to do is to understand the weakness of the bacteria. In other words, if you understand the enzyme, you can find a way of inhibiting that enzyme, the biochemical pathway. If you understand a particular reaction, we can intercept the pathogens process. So I'm going to describe in a layman's term few things. And basically we did the inhibitor development, and we developed the first set of killing inhibitors, and like this few things we did. I will not go into it. So what kind of enzymes we target? What kind of proteins we target? Just to tell you, TB bacteria is not very big. It's as, everybody has heard of a most well-studied bacteria called Escherichia coli, E. coli. Okay. More Nobel Prizes were working for E. coli in biology. Okay. When Delbruck, physicists, got into biology, started working on E. coli systems. Right. So it has about 4,000 genes, 4 megabase, 4,000 kb genome E. coli. So is MTB. It's similar. But that's the similarity ends there. Afterwards, many things are very different. Yet, the housekeeping functions are very similar. Housekeeping functions, the core architecture is very similar, which sustains the cell. For example, energy production by ATP, it's similar. Or many other things are similar. So we work on those essential enzymes needed for MTB. Of course, they are also found in E. coli or other bacteria, even in humans. But we try to choose such a way that they understand their properties. In what way TB bacteria has the different properties, subtle difference in their properties. So we work on these enzymes. I'll tell you what are these enzymes in a minute. And then we also looking at the genes. As you all know, not all the genetic makeup, every genetic component is expressed all the time. The DNA is static, RNA is expressed form, then finally proteins, there's a functional, right? So not all the genes are expressed all the time. Some genes are expressed sometime. So differential gene expression is the hallmark of life. If every gene is expressed all the time, then neither you will be sitting there, I will be standing here, right? So we work on this gene expression. The gene expression, the central enzyme is called RNA polymerase, DNA to RNA. We work on that. We also work on how when the gene synthesis transcription ends, the termination factor or other factors. So basically we target essential enzymes. We study these enzymes and try to find how to stop the activity of these enzymes. So I will not again go into all these things, just give one example. bacterial cell. This morning also I was interacting with the people, with the Krishnamurti, then Brato, and a little bit with the Rama. So basically, we are discussing some bacterial cells as well. Bacterial cell has a DNA. You don't have compartments, but DNA is compacted. It's not hanging around completely loose, highly compacted. And when you open up this, you can see this spread out of textbook. What makes the DNA compact inside the bacteria? The enzymes called topoisomerases, they compact the DNA, and also small proteins called nuclear associated proteins. Bacteria has no nucleus because there is no nuclear membrane, but the DNA, compacted DNA, is called nucleoid. So this nuclear compaction is by the action of these enzymes, these proteins. These are proteins, they are not enzymatic. They bind DNA, bend DNA, write DNA, twist DNA, and they make compact. Whereas these are enzymatic. How do they function? Just little, I will not go into details. These are two types of enzymes, topoisomerases. They are considered as the Mahapandit in this field called them magicians among magicians, simply because they bind the DNA, cut the DNA, either single strand or double strand, then pass the intact strand across, then the reseal. The chemistry, what they do is very fascinating. In between the reactions, they are covalently attached to the DNA. And phosphodiester bond is broken, then resealed. So basically, these are called type 1 because a single strand nick, intact strand pass through, then they are resealed. As a result, this whole duplex is opened up. The other way around is a duplex DNA cleavage, and then it's strand passage and resealed. So yes.
So this thing is compacting by itself, but I thought like DNA compacts by rolling itself around the histone and by some DNA.
That's okay. It's a good point. I'll tell you, histones counterparts are what I showed here. These are originally called as histone-like proteins in bacteria. And in fact, we work on them at this point. These are very fascinating bunch of small proteins. Many ways they're similar to histone, but they're different. See, histone, the eukaryotic chromosome is compacted by higher order structure. In the initial phase, they wound over the nucleosome. They wrap around the histone octamer. Then they come further, there's a higher order condensation. But there, in addition to topoisomerases, there are other, and histones, there are other proteins come into picture. We can discuss that later. This is more simple, yet it can be sufficiently complex. Basically, so these are called topoisomerases, and they solve all the topological problems inside the cell. And imagine, for example, when the genome has to be duplicated, the double-stranded DNA has to be opened up. And then the replication machinery has to multiply the copies, or transcription machinery has to transcribe a given gene. So DNA duplex has to be opened up. When you try to open up, there will be tangled mess. That is solved by this cleaving and resealing by the topoisomerases. For example, when the transcription, when the RNA synthesis is going on, DNA synthesis is going on, these enzymes have to work hand in hand. I don't have a slide here. And basically, then they are essential. If you inhibit them, cell death. That's why the topoisomerase inhibitors are magic drugs. Anti-cancer therapy, they are used. And your fluoroquinolones, when you go to ciprofloxacin, norfloxacin, the doctors give, is target DNA gyrase. Okay. Having said that, the topoisomerase one had no drug in bacterial kingdom. We discovered one. We are continuing. So as I said in the morning to Krishnamurti, I think I was talking to you about this knotted thing. Inside the cell, DNA gets highly compact. Whether it's higher organisms, bacteria, is not only compact, then can get knotted up, extremely tangled. Everything is sorted out by the action of topoisomerases. So if the inhibitors which target them act like a poisons. I'll be done in about next 10-12 minutes. Yeah. Yes. That's a good point. Krishnamurti is asking, you need treat. We are not worried about DNA. You're talking about the enzyme which works on DNA, right? It's a question of how different they are. In fact, I didn't go into the details. The bacterial enzymes have a distinct properties compared to the higher organisms. There are some properties which are similar, but you don't target them. I can elaborate again later on that. So, for example, this quinolone class of drugs, ciprofloxacin or related fluoroquinolones, they target an enzyme called denaturase, a type 2 topoisomerase, one which makes double strand breaks. Whereas eukaryotic topoisomers are targeted by something called etoposide, which is specific for eukaryotic one. The subtle biochemical differences. I can elaborate, but I think in the interest of general audience, I will not go into that. So, similarly, there is a plant alkaloid called camptothecin, and camptothecin is shown to be very effective against human topo 1, one which makes single strand break, with a very different kind of reaction there. And its derivatives are wonder drugs against certain cancers, even now. But that doesn't touch the bacterial enzyme because of the subtle differences in the way these transesterification reactions are carried out. Right? So, in this European Union consortium, where I was the only member from India, myself and my collaborator, what we did, we could find these small molecules. At that time, we didn't have a structure of the protein. Now we have the structure also. I collaborated with structural biologists. It could dock near the metal binding pocket. These small compounds are actually, it's a clinically used antidepressant. Way back in 1950s, it was used in TB sanatoriums in Swiss Alps, and people came out. But even now it's an antidepressant, but that time was it used as antidepressant. But what we found is it is docking nicely into Mycobacterium tuberculosis topoisomerase 1. Obviously, what I didn't say is we genetically manipulate, clone the enzyme, express the protein, we do all this biochemistry. I didn't go into the experimental part. So basically, if it works, actually, it's a case of repurposing, and that's what we have done. In fact, these days, every pharmaceutical company, everybody is looking into repurposing the drug because they developed for something else and put in the cold storage, it didn't work or whatever it is. So even metformin, which is used for diabetes now, people are thinking of anti-aging and in a very small dose, and and some of the other applications. You can look it up, and there are many repurposed drugs coming to things. So I will want to go into details. Basically, these compounds, so-called antidepressants, they kill the TB. And this is one of the essays. Blue means their cells are thriving, and purple means the cells are dead. And you can several log-fold killing of the bacteria. And we are pursuing now combination in the laboratory for this. With the first line rifampicin, RNA polymerase inhibitor, that kind of combination. I have a DBT project running with my last student. As in my prolonged, as I said in the lunchtime, prolonged death phase, we are trying to pursue this, and one of the last set of experiments in this. Now, I come back to this slide. Now I talk a little bit for a few minutes on this nuclear associated proteins. So we work on them from Mycobacteria. Mycobacteria has, again, compared to E. coli, very, very different. And some of them are characterized in my lab. And having had 40 plus 45 graduate students, many of my students have done wonders. I just come and give talk. So what we show here is, normally these architectural proteins are not essential. In MTB, this is essential. How do you know? Because you may make genetic tricks. You bring down the expression level. So this is one of the very few gels. We all, biochemists have to run the gel. Otherwise, you are out, not a biochemist. I'm a card-carrying biochemist. This is a protein gel. You stain the protein, called Western blots, right? After running. So what you see, the protein level is decreased. We have manipulated to bring it down. When you decrease the level, the cells are not growing, showing that this protein is important for cell survival. Am I making myself clear? Okay. MTB cells are not growing simply because you don't have enough protein. We can't knock out this. This is essential. That means it's essential gene, right? And then when you deplete it, just see this is a DNA staining. In the wild normal TB cell, in the conditional knockdown, it's a conditional knockdown because you're depleted gradually. The DNA is spread out. The blue color is a DAPI staining of the nuclear DNA spread out. So compact DNA has been spread out. So net result then what is happening here is, okay, basically I'll show you, given that, what we thought we can try to find drugs for this. So solve the structure is a dimeric protein and DNA sits in the saddle. There's a beautiful saddle kind of thing, DNA sits. So question is whether DNA can be thrown out by small molecules. And that's what we do. And by a lot of studies is in silico, we find these molecules are able to sit here and displace the DNA which was already sitting there. This is another kind of a DNA gel, radioactive DNA, and a DNA protein complex, this small nuke. And when you treat with the inhibitor, it comes back the free DNA. So basically, the small molecules are able to inhibit the protein function. With the result, I'll skip this slide. With the result, there is a cell killing, and the cells are deformed, and nucleoid is in a bad shape. That means it's a first proof of principle for targeting even the architectural proteins for drug treatment. Again, we are continuing that. But it also perturbs the overall DNA architecture and topology. So you can look at the overall gene expression changes. I will not go into detail. These are called one of those modern techniques, high throughput technology, transcriptomics, you call it. Okay, with the result, you see how a chemical perturbation of the genome can lead to large scale changes. So you see this small MTB protein affecting so many pathways. What is in blue, most of this gene expression, protein expression is downregulated when you treat with the inhibitor. That's why cells are getting killed. That's the point. So this is just an idea of last few minutes I'll say about something else. This is only part of the story. MTB has so many ways of dealing with the host, yet we don't know many things. Host has been evolving different strategies, immune evasion, many mechanisms. Now, I will talk about some new exciting developments, a tip of the iceberg, simply because MTB has few hundred proteins sent inside the host. Very unusual for a bacteria. It not only infects, after infecting, these proteins get secreted out of the host, get into the host. This is a recent science paper about 2 years ago. But independently, we have been working on MTB infection and looking at these proteins. I will not go. So, and many of these proteins are modification enzymes. They modify DNA or they modify RNA or modify proteins, the enzymes which modify. So when they modify the host proteins, nucleosomes, we talked about, then there's a host signaling can be altered, and that is their job. And that's what one of our recent study shows. There's one such TB protein which gets into the host, which not only gets into the host, it manipulates the host gene expression by methylating an enzyme, a protein called histone octamer. Then as a result, the host innate immunity is getting suppressed. and it adds its own mark to counter the host to oxidative stress and other stress. As a result, host controlled cell death called apoptosis is countered, and it's the bacterial winning bacteria kills the host. That's called necrosis. Necrosis happens. So all this kind of a double-edged manipulation is is carried out by the pathogen, and this is summarized in this figure. Basically, an enzyme called 2067 gets into the host, gets in the cytoplasm itself, it methylates histone H3, and inside it does many things, basic and increasing necrosis and decreasing apoptosis by multiple mechanisms. This is we uncovered. We were trying to find out more things about it. Just wanted to show you, and even this can be used for a new drug strategy. And in fact, there are few more such proteins which get into the host doing different things, which you have compiled here, but I won't go into the details. Each one is doing different way. In other words, there are multiple strategies by the pathogen to outmaneuver the host. When the host is trying its own, in the co-evolutionary arms race, this is what we discovered, and I have shown here. The rest of it is from different people. I will not go into these details because it's not because slide was there, but only this part. But anybody is interested, I can elaborate on any one of these things. So basically, MBT and MTB are very similar in words, just one word interchanged, but main battle tank can be destroyed easily, but MTB cannot be destroyed that easily. That's the point. And the final slide. What you need to treatment, as I said, if somebody can find a drug which can treat TB in 1 month, that's a dream of many people. People are passionate in working in that area, but it's not going to be that easy, as I said. So you have to have completely sterilizing bacterial activity. Oral delivery will be preferable. Effective against persister cells. Persister is the dormant latent TB cells. Novel mechanism of action, and there should not be antagonism. See, when you treat with a few drugs, they cannot start creating a problem between them. Compatible dosing, compatibility for antiretroviral. Antiretroviral means HIV kind of therapy, and toxicologically acceptable dosing. Even 2 months is fine. Short is a big challenge. The barriers, of course, fine. See, it's not a lifestyle disease. If it is a lifestyle disease, there is money in all these rich countries. And this is a poor man's disease. Financial incentives are inadequate in most, in fact, even the major companies like Novartis, and they're all backed out. They are working at TB at some time or other. They have abandoned the programs. Pfizer withdrawal, AstraZeneca, Novartis pull out. So limited financial support to progress clinical drug candidates in the public sector. Even we had some fantastic leads way back 15, 20 years ago. The Indian pharmaceutical company Lupin, institute was not patenting that time. It had more than 50% of the rifampicin market. So they did the PCT patent for me. I mean, it was a TDB joint project, but afterwards they were not interested in taking it further. This is the situation, because they were looking for somebody else to buy it or something like that. So this is a situation. So I thank you for your attention, and I try to avoid giving the data, and I do only the talking and pretend to. In fact, team members are rowing the boat. Thank you.
Thanks for a nice talk. I was wondering, in other animals you mentioned, like in cows or in many other mammals, probably also in probably even primates, other primates.
Yeah, absolutely.
So is the
in fact, monkey models are used for infection. See, what happens? Sorry to interrupt because I wanted to answer this point. When you do the rodent is a animal model, what is true in rodents doesn't apply. In many times the clinical trial things fail because of such situations, and people put a lot of money into research, and then the physiology is so different, doesn't work. So there's a primate model is very expensive, but people work on that before bringing into the market many things. So there's the MTB what infects humans and the MTB which infects primates are very similar. Yeah.
Are very similar.
Yeah.
But not for, say, cows or rodents or anything like
Cow is also less than 1% difference. In fact, Mycobacterium bovis and MTB has very little difference. That's why I said they are all. You see, originally it must have come from other primates to humans. The TB bacteria.
So I was just wondering, firstly, so you said these bacteria have co-evolved also with the organism, so that's why probably there is a variation. But is there some strategies that some other animals have evolved to combat their infection, which maybe we could...
it is a fantastic point.
Is the incidence, for instance, of TB in other animals as much as it is in humans? It's 1/3.
You don't get to know that much. In fact, what is thought about in India, it's a possibility exists. It's not as severe in, but I know of people these days, especially when you go to the vets, the dogs and died of a tuberculosis infection. So with a wild animal, you don't know this what's happening, and perhaps they have more robust immune system. That's a very good point what you raised, but everything gets seen in our context. So your point is very relevant when it comes to your COVID virus in bats. Bats it doesn't come down with the disease, but they are good residents. So is your so many mosquito-borne diseases. Nothing happens to mosquitoes. Of course, they are invertebrates. It's a good thinking, but then how do you study there? Question: if it's not manifested, if the TB bacteria likes to hide there, yeah. But it's not uncommon, I must tell you, to get the animal TB infected into humans. So these days, what we're talking about is not only tuberculosis. There's a tuberculosis complex. There's something called Mycobacterium avium, bird TB, and Mycobacterium marinum, that is fish TB. That is not there because you eat the fish. That doesn't happen that. But with many other related to mycobacteria, in fact, 15% of the infections, I just learned in last week in the conference, is a non-TB bacteria which causes TB, but related, the large TB subfamily. Coming from, from in India, in village settings, you have a close contact with the animals, right? Yes. Yeah, you have close, so it's unavoidable such infections. So it's a, yeah. 15. Poultry farming, cows, dogs, cats. Yeah. And there are some of these TB bacteria can infect many things. Even Mycobacterium bovis can infect humans. Yeah. See, the BCG is Mycobacterium bovis BCG. This is very close. Yeah.
I had a question in general about combating like drug-resistant bacteria, maybe not just TB, but I recently came across people trying to look at how the bacteria survives not just in the host, but in the microbiome of like the host. So maybe
which one? TB bacteria.
Not TB in general, but like any bacteria. Apart from its like
microbiome. Okay,
yeah,
in my microbiome.
Yeah, and perhaps utilizing how the bacteria may be like strong on its own, but in the microbiome, it doesn't have a very good survival rate. and maybe utilizing those kind of strategies to combat. So
For
basically,
TB,
not like
for TB bacteria, I mean.
I'm just asking something like that.
Yeah, that's a good point, but I must. When you talk about microbiome, you can talk about microbiome of, I teach microbiology. I have taught microbiome, so I can give a long answer, but I'll keep it as short as possible. Is microbiome can be at different place.
Right, not
in the skin, inside the stomach, inside our oral, in our mouth, in different urinary tract. Name a place, there is a microbiome.
Yeah.
Right? What we do in the morning is we get rid of 0.1% of the bacteria by scrubbing different, irrespective of the soap. We scrub only, and that's still most of the bacteria there. We have more microbial cells in our body, estimated either 10 times or 100 times, different estimates are there. But we only get rid of a part of it. When you say microbiome, there's a huge number of microbial consortium, let us say gut microbiome. That is the most well studied. But there, the population varies, but you don't have MTB so much. Yes, there is a stomach TB under those conditions. Why it is not curtailed? That's an interesting question, but so far in the microbiome literature, I don't see those kind of things. Microbiome option treatment is thought about for a few things. But here, as I said, the MTB bacteria is very formidable. It has found a way to survive itself under very hostile situation, right? Not many bacteria can do that. There are a few other bacteria which can do. So given that, in microbiome, you must also realize many of these bacteria are the population numbers are very different. Some of them are multiplying, some of them are more, what shall I say? They are in a smaller number. They don't multiply that fast. In fact, some of them cannot be cultured in the laboratory. Some of these microbiome bacteria, only few can be. Of course, we keep improving the technologies because they're called unculturable bacteria. So I'm not sure that's a good option, because TB infects into lungs, and in the lung, suppose the same time if you have some other lung infection, something else, they are getting cleared, but TB is not getting cleared. But that's where the situation also, when you have suppose if you have 2 or 3 organs at the same time, they can further aggravate the situation. Under those conditions, I don't know how you can control the TB bacteria. I hope.
No, I was not even sure that TB even interacts with your microbiome. And if studies are being done about how it's
No, there is not done that way, because TB bacteria has many niches. See, in one sense, TB bacteria, what it has its own niche in so many things, We are, in the skin TB, when you call about, or skeletal TB, it need not necessarily interact with other microbes. But at the same time, I agree with you, there may be a point in some cases, the resident microbes may counter the TB. But that question has been not addressed. It's actually a very beautiful point. I don't have a direct answer, but this is quite thought-provoking. But no, there is no study as of now. But as such, you don't inject TB bacteria into rat stomach and look for the microbiome clearing, or I haven't seen that kind of a paper yet. Yeah.
Thank you.
Yeah. But microbiome is not solution for everything, I must tell you. Yeah. Yeah. Very interesting point. Interesting point, yeah.
You said that the drugs that are available.
You may want to turn it on, yeah.
So you said that the drugs that are available for TB do not work on the dormant MTBs
Yes.
and only on the active ones. So, like, what is it that these drugs are targeting, which is absent in the dormant ones but present in active ones? Which
No, as I said, the dormant bacteria, we know it is there because you see the reactivation. But we don't visualize there is no technique you are able to visualize the dormant bacteria. You found a few, some stem cells, they found bacteria hiding, but they've seen the reactivation happening from those cells, but they have not really visualized a clear dormant state. There are evidences, but not strong enough. So given that, and also they're recalcitrant. So the drugs have to get in there to target them, even if you find. That has been remains a challenge at this point. When it is active TB, then bacteria can get into the TB and treat, right? Even if it is inside the, when somebody has a TB, then when you give the injectable drugs get into the cells, then get into the host, get into the bacterial cell and inhibit these enzymes, right? Either they break the bacterial multiplying cell wall or inhibit the transcription machinery or replication machinery. In the dormant bacteria, they are not multiplying, they are dormant. So you can't, the gene expression is the lowest level. So that's another challenge.
Okay, so if not, let's thank Professor Nagaraja.
Thank you.